Hi guys, welcome back to the channel. In this video, I will be covering the enzymes topic of the A-level biology curriculum. Let's begin. So enzymes are biological catalysts. So as you might be aware that catalysts speed up the rate of reaction. So being biological catalysts, they basically speed up the rate of reaction in living things or living organisms. And they do that by lowering the activation energy of the reaction they catalyze. So they would work by lowering the activation energy. Activation energy is the minimum energy required to initiate a reaction, and they basically lower that, so a reaction will need less energy than it would usually need, and that's how they work. And how they lower the activation energy, so what they do is they bend and or they distort the bond. So this is a bit different to what you might have learned in chemistry, but this is what you need to know for biology. So they bend or they distort bond, and that's how they lower the activation energy of the reaction. So you need to be aware of the specificity of the enzyme. So enzymes have a specific tertiary structure and active site. So if you remember from the proteins topic, the tertiary structure is the 3D structure, which comes after the primary and the secondary structure, and the enzymes have a specific tertiary structure which no other protein will have. So it has a specific structure and a specific active site. Active site is where the substrate will bind to. So the active site is complementary to the specific substrate. So complementary is a keyword that you should learn and make sure that you are using it whenever describing the relationship between the active site and the substrate. So the active site is complementary, which means that it binds perfectly to the substrate, which will be specific to the active site. And the only the specific substrate, so the specific substrate can bind to the active site of the enzyme. So if it's not really any substrate that can bind to a given active site, it will only be a specific substrate that can bind to the active site of the enzyme. So for example, in this case, we have a substrate and an enzyme. So this substrate is complementary to this enzyme and will bind to this. If I just get a random substrate and try and bind that with the active site, this won't work, because I need the complementary substrate which will bind to the active site of the enzyme. And the properties of enzyme relate to the tertiary structure of the active site. So this active site here indicates the properties of the enzymes and what role the enzyme will have in the body or anywhere. So it's the active site that's the important part of the enzyme. Alright, so you need to be aware of the lock and key model. So what this indicates is that the active site has a fixed shape. So in this example, the substrate and the enzymes would so the substrate would exactly fit into the enzyme, and the enzyme active site shape does not need to change, and it will not change. And the active site is complementary to the substrate. So as you can see, the active site has the shape which the substrate will be able to bind to, and that's what makes it complementary to the substrate. And after successful collision, an enzyme substrate complex forms, and this is the enzyme substrate complex. So this was the lock and key model, but this is not used anymore, and the model that's used now is the induced-fit model. So in this, what's indicated is that before the reaction, the enzyme active site isn't complementary to the substrate. So for example, in the picture we can see is that the active site has this kind of shape, but the substrate has a slightly different shape. As you can see, it's still similar, but it's not exactly the same as what would be shown in a lock and key model. And so what happens is that when the substrate binds, so in this example, when the substrate binds, the active site changes shape to make the substrate complementary to the active site. So the enzyme changes its shape, and this is how a enzyme substrate complex is formed. And this bends or distorts the bonds, leading to a reaction. And after the reaction has finished, the enzyme's active site returns to the original shape. So in this, the shape changed when the substrate was binded with the enzyme, but after the reaction finished, the enzyme, as you can see, returned to its original position, and this cycle would just repeat, and the enzyme would change when a substrate binds. The enzyme would change the shape again to make it complementary, and then it would return to its original position, original shape when the reaction has finished. Right, so how the temperature affects the rate of enzyme controlled reaction. So increasing the temperature increases the rate of reaction. So as you can see in the picture, as we increase the temperature, the rate of reaction increases. Now this is because there will be increased kinetic energy. So particles are moving around faster, and this means that there will be more successful ES collisions, which is the enzyme substrate collisions. You can also call it ES. And therefore there will be more enzyme substrate complexes being formed, and which basically lead to the reaction occurring. But increasing the temperature above the optimum temperature decreases the rate of reaction. So increasing the temperature increases the rate of reaction until a point which we call the optimum temperature, the optimum point. So this is where the rate of reaction will be the highest. But after that, as we increase the temperature, the rate of reaction starts to decrease. Now this is because the enzyme active site denatures. So the tertiary structure and the active site change in shape. So make sure that you learn this keyword denatures, and you use that when you're talking about the active side of the enzyme changing shape. So you use the word denatures instead of saying destroying or anything else. So this is because the hydrogen and the ionic bonds in the enzyme start to break, which changes the structure, and the substrate is not complementary to the active site anymore, so it will not bind to the active site, and therefore fewer successful enzyme substrate collisions occur, and therefore fewer ES complexes formed, so it slows down the rate of reaction. So this is the enzyme. So this would be the normal enzyme, and this would be the substrate binding to it. But when the enzyme is denatured, as you can see, it changes the shape of the enzyme. So the substrate, if it was to bind to that, it would not be able to, as there has been a change in the active site, and the enzyme active site is no longer complementary to the substrate that it was complementary to before. Alright, so how the pH affects the rate of enzyme controlled reaction. So pH, if you change the pH below or above the optimum, so if this was the optimum and you change the pH below, or you increase the pH, it basically decreases the rate of reaction. And this is because the enzyme denatures, and again the ionic bonds. So this time ionic bonds, because remember the pH is basically the concentration of H plus ions. So H plus ions, and this is a example of a cation, and they would form bonds with negative ions, which would be the anions, and they would form ionic bonds. So changing the pH would alter the concentration of these hydrogen ions, and so this would interrupt with the ionic bonds and denature the enzyme, and therefore the tertiary structure and the active site of the enzyme change shape. And again, the substrate is no longer complementary to the active site. Again, the same occurring in this instance. And again, so fewer ES collisions, successful enzyme substrate collisions, and therefore fewer enzyme substrate complexes being formed, so there will be a slower rate of reaction. So how does the substrate concentration affect the rate of enzyme controlled reaction? So increasing the substrate concentration increases the rate of reaction. So there is the positive correlation. This is because the substrate concentration for now is the limiting factor. So it's the substrate concentration which needs to be increased for the rate of reactions to be increased. So more successful ES collisions form, and ES collisions will occur when the substrate concentration is increased. So there will be more ES complexes, enzyme substrate complexes being formed. And the rate of reaction would start to plateau after, as you can see, after this point. And now this is because the enzymes concentrate, because now you've added enough substrate, but now the enzyme concentration is starting to become the limiting factor. So it's not the substrate concentration anymore. If you want to increase the rate of reaction further, it would have to be the enzyme concentration that you should be looking at now. Right, so how does the enzyme concentration affect the rate of enzyme controlled reaction? Now this is very similar to the substrate. So what happens is that increasing the enzyme concentration would increase the rate of reaction, and this is because again enzyme concentration is the limiting factor this time, and if there are more enzymes, there will be more active sites available for the substrate to bind to, therefore more successful ES collisions and more enzyme substrate complexes being formed. And the rate of reaction, again in this example, it starts to plateau after a certain point. This is when the substrate concentration would become the limiting factor. So to increase the rate of reaction further, it would have to be the substrate concentration that you would have to increase. Now, how does the competitive inhibitor affect the rate of enzyme controlled reaction? So adding a competitive inhibitor decreases the rate of reaction. So this is because the competitive inhibitor has a very similar shape to the substrate, and so as you can see in this picture, and this is the substrate, and the competitive inhibitor has a very similar shape to the substrate. And what the competitive inhibitor does is it will bind to the active site of the enzyme. So this is the active site of the enzyme, and it blocks the active site. So if the substrate was to come now, it will not be able to bind because the active site is blocked by the competitive inhibitor. So what this causes is there will be fewer enzyme substrate complexes being formed, but this inhibition can be overcome by adding more substrate. So if I was to add more substrate, then the decrease in rate of reaction could be lowered. So this can be overcome by adding more substrate. So in this, the reaction is much slower than what it would usually be, but the maximum product is still reached. So you still form the maximum product that you would normally form, but this would form much slower. We'll be looking at a graph later to help with your understanding. So how the non-competitive inhibitor affects the rate of enzyme controlled reaction. So in this, again, it decreases the rate of reaction. So the non-competitive inhibitor, what it does, it binds to the site away from the active site. So it's not the active site it's binding to, it's binding to the allosteric site. So this is the non-competitive inhibitor in this example, and this is the active site. So it's not binding to the active site, it's binding to a site away from the active site, which is the allosteric site. But what this does is it changes the active site's shape. So it would bind to the allosteric site, and this would ultimately change the active site of the enzyme, so the substrate can no longer bind to the enzyme. So it changes the shape of the active site, so the substrate is no longer complementary to the active site. So again, there will be fewer enzyme substrate complexes being formed, and in this example, increasing the substrate concentration has no effects on the rate of reaction. Now this is because this is affecting the enzyme, so the enzyme would not be able to form any substrates this time. So again, therefore there will be no effect on the rate of reaction if you increase the substrate concentration. And in this example, the maximum amount of product will not be reached like it was reached with competitive inhibitors. So looking at this graph, as you can see, the orange one is competitive inhibitor, red one is non-competitive inhibitor. So the grey one would be the normal without any inhibitors. So this would have, the grey one would have the fastest rate of reaction. Orange one, it has a slower rate of reaction, but it does form the maximum product as the lines, the grey and the orange lines meet. But the non-competitive inhibitor, as you can see, it does not form the maximum product. It plateaus much earlier, and the reaction would stop much earlier, and then in both of the competitive inhibitor and without the inhibitor. Thank you so much for watching this video. If you did like this video, don't forget to subscribe to see more of these, and you can watch my recent videos by clicking on the links popping up. Thank you.